Well, hello students, and welcome to this first recorded lecture for your cell biology module of BCH 1118. So this video is for students studying pharmaceutical science and will be the first part of the cell biology material for your introductory biochemistry course. Okay, so I want us to start thinking about studying cell biology, the biology of cells. So the first thing I want you to think about is, what is a cell? I'm sure that most of you have already learned quite a lot about cells. Hopefully you have studied biology at A level and at O level, so you will have learned about cells. So I'd like you just to pause the video for a few minutes and to draw or write, or if you have a friend there with you, you can talk about it, what you can remember about cells, what you've already learned. This is just to help you remember what you already know. It's not intended to be a test or to be anything difficult, just to help you to remind yourself what's already there in your knowledge. Okay, so just pause for a minute now. Okay, so what is a cell? Well, a cell can be defined as the basic unit of life. So a single cell can be alive. There are many, many single cell organisms on Earth. So if I'm talking about a cell being alive, what do I mean by life? Again, I hope that this is something that you will have already learned, how life is defined. So if you remember that, good. If you have forgotten, please do look it up. Check on the characteristics of living organisms. Make sure that you know what I mean by saying a cell is alive. Okay, so a single cell can be alive. Multicellular organisms are made up of many cells. A third category that you might have come across is viruses. You will have heard a lot about viruses, about the COVID virus recently, for instance. There is a lot of discussion, there is some disagreement about whether we should say a virus is alive or not. Personally, I think that viruses should not be classified as being alive, because viruses are not able to reproduce themselves without using the machinery of a living cell. Other people disagree with that and say that viruses are alive. So you might want to think about how you would define a virus. Is it alive or is it not alive? Okay, I'm going to pause there and let you think about any questions you might want to ask. You can write them down. And if you want to address questions to me, please do use the discussion forum on the LMS. Okay, so thinking about cells, the next question I want to ask is, how big is a cell? And to think about this, I'm going to show a video. Okay. Okay, I hope you understood what was going on there. So we saw a video zooming in, if you like, from things that we're familiar with. So here is a coffee bean, here is a grain of rice and a sesame seed. And up here in the top hand corner is a piece of squared graph paper, if you like, with this square representing 1 millimeter square. So you know how big a grain of rice is, for instance. Here is some print times 12 point print, a very common font that's used. You know how big that is. So we're starting in the macroscopic world. Macroscopic, a word meaning things that we can see with our naked eyes. So you can see here, you know that a sesame seed is quite small, but you can see it quite easily. So we're zooming in on things that are smaller. So if we go now to the next slide, we can see the largest cell that we know about. So this is an amoeba proteus. It's a single-celled organism, but its diameter is about 0.5 millimeter. It is big enough to see with the naked eye. This single cell is just about big enough to see without any magnification. You can just see a tiny dot moving around. Here is a paramecium, another single-celled organism. And here is a human egg cell. Okay, so this would be really just on the edge of being able to be seen without magnification. Zooming in even more, we can see a skin cell. So this is about 30 micrometers across. Here's the human egg again. So we've zoomed in, so it's looking bigger. There's the egg, and here is the sperm that will fertilize this egg, much, much smaller. Here is a specialized human cell, a photoreceptor, very long, but very narrow. So 2.5 micrometers by 100 micrometers. Zooming in some more, we can see a red blood cell, so much smaller than a skin cell, for instance. So here's the red blood cell. And here is another single-celled organism. This is Baker's yeast. And you can see that this is much smaller than a red blood cell. So we're seeing a big range in size of cells. And here we're starting to see things inside cells. So here is a chromosome from a human nucleus. Zoom in again. And here is another single-celled organism, E. coli bacteria. And here, for comparison, is an organelle within a eukaryotic cell. Here is a mitochondrion, and here is another organelle, a lysozyme. So you can see that E. coli is quite small and of a similar size to a mitochondrion. Okay, so I'm going to go back and play the whole video again, and hopefully you'll be able to understand it a bit better now that I've explained it to you. So zooming in, a grain of salt, and there's the big amoeba cell, paramecium, a skin cell, a red blood cell, an X chromosome, E. coli and a mitochondrion. There's a measles virus and some other viruses. There's a ribosome, an antibody. And here we're down to small molecules and a single carbon atom. Okay, so I hope that that helped you just to picture how big a cell is and the large range of sizes that cells can have. Okay, so we've seen different sizes of cells. Now we're going to see different shapes of cell. If you drew a picture of a cell when I asked you to earlier, I think you might have drawn something rather like this, something fairly round with a nucleus in the middle. Perhaps you remembered to put some other organelles in it as well. Perhaps you remembered to put a membrane around the outside. Perhaps you even drew a plant cell and put a cell wall around the outside. But even though we tend to think of this as a typical cell, cells come in so many different shapes and sizes. So here is just an illustration of some different cells. So here is a liver cell, which is not so dissimilar to what we looked at, a fairly round cell with a nucleus in the middle. But what about this? I wouldn't think that anybody would draw something like this if they were drawing a picture of a cell. This is a nerve cell with a long extended axon making contacts with lots of other cells. Here is another type of nerve cell. This is the sort of nerve cell we might find in the peripheral nervous system, whereas this is the sort we might find in the brain. So a more round structure, but again making contacts with lots of other cells. Here's a human sperm cell, a very specialized cell again. Here are some single-celled organisms, some bacteria. Here is a Chlamydomonas, a blue-green algae. You can see just by looking that there are lots of different shapes and sizes and functions of cells. So when we look at a cell, we can see how their shape and their size reflect what sort of cell they are, what the cell does. Okay, so another little video just showing you some different cells, just to put it into an attractive video to watch. Okay, there's a virus, a bacteriophage, injecting its genetic material into a cell. There's a dividing cell. There's a nerve cell making contact across a synapse. Here are some bacterial cells moving. Here are two cells fusing. And a multicellular organism like a human being has literal trillions of cells as part of that organism. And they make up our bodies. Here are cells with projections on the outside, and here's a very round, smooth cell. Here's a cell again with projections, lots of cilia on this cell. Here's a cell with two nuclei, probably about to divide. And here's a network of cells making contact in a nervous system. Cells releasing things. That's an oocyte being released from an ova. There's a brain stem and all the cells forming the brain. Red blood cells in a vessel. And a human being, a multicellular organism made up of many different types of cell, even though they all make up the single human organism. Okay, so if we look at all these different sizes and shapes and functions of cells, we might become confused about exactly what we do mean by a cell. However, even though they look so very different, there are a number of things that these cells have in common. So here is a picture of a real cell. Most of what I've shown you so far has been illustrations of cells, but this is an electron micrograph of a single cell. So this is a human cheek cell, which is an epithelial cell. And we're looking at a picture taken using a light microscope, using a technique called phase contrast microscopy. So the cell is magnified about 400 times. So just looking at this picture, you can see that the cell is a defined unit. It's got an outside, it's got a boundary around the cell, and you can see there's a distinct difference between the inside of the cell and the outside of the cell. Inside the cell, you can see that there are things inside the cell. So here is a structure which is the nucleus. Here you can't see much detail, but you can see that there are other structures within this cell. So here is another cell that we're looking at in more detail. So this is a single-celled organism called Chlamydomonas, which is a blue-green algae, a type of plant cell, if you like. And here we're looking at a photograph of the cell seen using an electron microscope. So electron microscopes give much higher resolution. We can see lots of structures inside the cell. So here is the nucleus inside the cell, and here is a membrane surrounding the nucleus. Here is another structure which is actually a vacuole, and lots of structures around which are made of membrane. Membrane-bound structures here with little circles inside them. This is actually a little circle of DNA inside a chloroplast. Remember that this is a plant cell. So here are chloroplasts with little circles of DNA inside them. So we can see that there's a high degree of organization within the cell and many distinct structures. The cell is not at all random. It's very carefully put together. Okay, another pause where you might want to just pause and write down any questions that you want to ask about what we've just learned. Okay, so here again is the picture of, if you like, a generalized cell. And in this picture, we can see indications of organelles within the cell. Organelles is the name given to the structures inside cells, a bit like organs inside the body, which are structures which are separate from each other. So the heart is separate from the liver, which is separate from the lungs, and so on. And each of the organs has its own specific function. Well, in the same way, each of the organelles within a cell are separate and distinct from each other, and each has their own function in making the cell work. So here we can see the nucleus of the cell. Here is endoplasmic reticulum, smooth and rough. Here are mitochondria. Here are centrioles, which are involved in cell division. Here is the Golgi apparatus for processing proteins and other molecules. A lysozyme for recycling, getting rid of waste and recycling it. Here we can see some hints of the cytoskeleton, which gives the cell structure. So these are microtubules. And little dots, which are ribosomes, producing proteins in the cell. So we'll be looking at all these different organelles next time and seeing exactly what they do and how they do it to some extent. Okay, so that's just a definition of what I mean by organelle. A subcellular compartment or large macromolecular complex, often membrane enclosed, but has a distinct structure, composition, and function. So another point that's important to remember is that not all cells have all the possible organelles. So there are even some cells without a nucleus. Mature red blood cells, for instance, have lost their nucleus, although immature red blood cells do have a nucleus. Some cells are packed with mitochondria for producing lots of energy. Other cells have just a few because they don't need so much energy. Not all cells have a cytoskeleton. So not all cells have all the possible organelles. So we've seen so many different sizes and shapes and functions of cell, but there are two basic types of cell that's important for you to know about. Two basic types of cell, prokaryote and eukaryote. Sometimes it's spelt with a C rather than a K. You might see that spelt that way. It means the same thing. So prokaryote cells and eukaryote cells. So these words come from the Greek words meaning karyon, which means a kernel or a nucleus, and the prefix is eu, which means true, or pro, which means before. So these two different types of cells were named thinking they represented before nuclei and true nuclei. Excuse me. Okay, so prokaryote cells are always single-celled. There are no multicellular prokaryotic organisms. So a prokaryote is a single-celled microorganism whose cells do not have a well-defined membrane enclosed nucleus. Whereas a eukaryote, so a prokaryote can be either a bacteria or an archaea. Archaea is a group of life that is somehow intermediate between prokaryotes and eukaryotes, but archaeal cells also do not have a, a nucleus. And the second type of cell is a eukaryote. So a eukaryote can be a single-celled organism or a multicellular organism. So an organism composed of one or more cells that do have a distinct nucleus. So prokaryote do not have a nucleus. So they're either a bacteria or an archaea. Eukaryotes do have a distinct nucleus. Okay, so here is a generalized picture of a prokaryote cell, a bacteria, a typical bacterial cell. So here is the genetic material, the DNA, which is loose in the cytoplasm. There is no membrane around it. There are also no membrane-bound organelles. There are no mitochondria or lysozymes or anything like that. There is a cell membrane around the outside of the cytoplasm of the cell, And many bacteria also have a cell wall, which is represented by the red structure here. So prokaryotes have no nucleus and no membrane-bound organelles. They tend to be small cells and simple cells. They're single-celled organisms, either bacteria or archaea. So again, a generalized picture of a eukaryote, a typical animal cell. So here we do see a nucleus with a membrane around it, a nuclear membrane. And going with that, other organelles with membranes around them. So here are mitochondria, here is endoplasmic reticulum, here is the Golgi apparatus. So eukaryotic cells tend to be larger than prokaryotic cells, more complex, and can either be an organism by themselves or part of a multicellular organism. So all the living things you see around you, animals, plants, small things like yeasts, algae, fungi, many, many more things that we're familiar with are eukaryotes. So this is a typical animal cell, here in the same way as a typical plant cell, also a eukaryote with a membrane-bound nucleus. Some differences to an animal cell in that there is a cell wall around the plant cell, and here we see a large vacuole within the cell. Okay, so if we compare eukaryotes and prokaryotes, we can see that they do have quite a lot of similarities. They are both types of cell, so they do have some features in common. So both types of cell have a cell membrane, that's the outside of the cell here, surrounding the cytoplasm. Okay, so here on the prokaryotic cell, and here on the eukaryotic cell, sorry, it's the black layer on the prokaryotic cell, and the red layer of the eukaryotic cell is the cell membrane, which is a lipid bilayer. We'll learn a bit more about that later. Both have genetic material, which is DNA, which carries the instructions to make everything else in the cell. So nucleoid here in the prokaryote is the DNA, and inside the nucleus in the eukaryote is DNA. Both have ribosomes, which are the structures within the cell that make proteins. So we'll be learning more about proteins and ribosomes soon. Both types of cell have cytoplasm, which is the aqueous material inside the cell, which is made up of water and lots of things dissolved in that water. Okay, so lots of things are similar between eukaryotes and prokaryotes. They also do similar things. Both types of cell can reproduce themselves. They can copy their DNA, a process called replication, which we will learn a lot more about. They can transcribe and translate the information carried in the DNA. And the molecular machinery that they use to do that is similar, it's comparable, although it's not identical. Both types of cell have similar needs. They both need energy to live and grow and reproduce. And to obtain that energy, they have to get it from a number of different sources, chemical and physical sources. So they both have a need for energy and to make use of energy sources. Okay, another pause for you to think about questions. You may want to pause the video again and write down anything you want to ask. So now I'm going to move on to thinking about how we know these things. How do we know about the structure and function of cells and organelles? So if we think historically, first of all, we found out that living creatures are made of cells, and the development of cell theory, then techniques to look at cells, to image them using microscopes and other magnifying instruments like electron microscopes, staining to let us see what's inside a cell, separation techniques to separate cells from each other and separate organelles within the cells from each other, and most recently, high resolution techniques, which allow us to see cells and structures within cells right down to an atomic level. Okay, so another little video about cell theory. What do we mean by cell theory? This is quite an amusing video. I hope you enjoy it.
One of the great things about science is that when scientists make a discovery, it's not always in a prescribed manner, as in only in a laboratory under strict settings with white lab coats and all sorts of neat science gizmos that go beep. In reality, the events and people involved in some of the major scientific discoveries are as weird and varied as they get. My case in point, the weird history of the cell theory. There are three parts to the cell theory. One, all organisms are composed of one or more cells. Two, the cell is the basic unit of structure and organization in organisms. And three, all cells come from pre existing cells. To be honest, this all sounds incredibly boring until you dig a little deeper into how the world of microscopic organisms and this theory came to be. It all started in the early 1600s in the Netherlands, where a spectacle maker named Zacharias Janssen is said to have come up with the first compound microscope, along with the first telescope. Both claims are often disputed, as apparently he wasn't the only bored guy with a ton of glass lenses to play with at the time. Despite this, the microscope soon became a hot item that every naturalist or scientist at the time wanted to play with, making it much like the iPad of its day. One such person was a fellow Dutchman by the name of Anton van Leeuwenhoek, who heard about these microscope doohickeys, and instead of going out and buying one, he decided to make his own. And it was a strange little contraption indeed, as it looked more like a tiny paddle the size of a sunglass lens. If he had stuck two together, it probably would have made a wicked set of sunglasses that you couldn't see much out of. Anywho, once Leeuwenhoek had his microscope ready, he went to town looking at anything and everything he could with them, including the gunk on his teeth. Yes, you heard right. He actually discovered bacteria by looking at dental scrapings, which, when you keep in mind that people didn't brush their teeth much, if at all, back then, he must have had a lovely bunch of bacteria to look at. When he wrote about his discovery, he didn't call them bacteria as we know them today, but he called them animalcules because they looked like little animals to him. While Leeuwenhoek was staring at his teeth gunk, he was also sending letters to a scientific colleague in England by the name of Robert Hooke. Hooke was a guy who really loved all aspects of science, so he dabbled in a little bit of everything, including physics, chemistry, and biology. Thus, it is Hooke who we can thank for the term the cell, as he was looking at a piece of cork under his microscope, and the little chambers he saw reminded him of cells, or the rooms monks slept in their monasteries. Think college dorm rooms, but without the TVs, computers, and really annoying roommates. Hooke was something of an underappreciated scientist of his day, something he brought upon himself as he made the mistake of locking horns with one of the most famous scientists ever, Sir Isaac Newton. Remember when I said Hooke dabbled in many different fields? Well, after Newton published a groundbreaking book on how planets move due to gravity, Hooke made the claim that Newton had been inspired by Hooke's work in physics. Newton, to say the least, did not like that, which sparked a tense relationship between the two that lasted even after Hooke died, as quite a bit of Hooke's research, as well as his only portrait, was misplaced due to Newton. Much of it was rediscovered, thankfully, after Newton's time, but not his portrait, as sadly no one knows what Robert Hooke looked like. Fast forward to the 1800s, where two German scientists discovered something that today we might find rather obvious, but helped tie together what we now know as the cell theory. The first scientist was Matthias Schleiden, a botanist who liked to study plants under a microscope. From his years of studying different plant species, it finally dawned on him that every single plant he had looked at were all made of cells. At the same time, on the other end of Germany was Theodor Schwann, a scientist who not only studied slides of animal cells under the microscope and got a special type of nerve cell named after him, but also invented rebreathers for firefighters and had a kickin' pair of sideburns. After studying animal cells for a while, he too came to the conclusion that all animals were made of cells. Immediately, he reached out via snail mail, as Twitter had yet to be invented, to other scientists working in the same field, and it was Schleiden who got back to him, and the two started working on the beginnings of the cell theory. A bone of contention arose between them, as for the last part of the cell theory, that cells come from pre-existing cells, Schleiden didn't exactly subscribe to that thought, as he swore cells came from free cell formation, where they just kind of spontaneously crystallized into existence. That's when another scientist named Rudolf Virchow stepped in with research showing that cells did come from other cells. Research that was actually, how to put it, borrowed without permission from a Jewish scientist by the name of Robert Remak, which led to two more feuding scientists. Thus, from teeth gunk to torquing off Newton, crystallizations of Schwann cells, the cell theory came to be an important part of biology today. Some things we know about science today may seem boring, but how we came to know them is incredibly fascinating. So if something bores you, dig deeper. It's probably got a really weird story behind it somewhere.
Okay, well, I hope you enjoyed that little video, and I hope that you also learned a little bit about the history of cell theory and how we know what we currently know about cells. Okay, so here is just a timeline putting this information in context. So here is Robert Hooke looking at cork cells through a microscope, his early microscope. Here's Leeuwenhoek discovering bacteria and protozoa. Here is Schleiden and Swan proposing the cell theory in 1838. And from this point, we started making discoveries very, very quickly. Staining to help us see different things inside the cells, more types of living organisms being identified, so bacteria being found to be responsible for diseases such as TB and cholera in 1882. 1931, an electron microscope, so seeing more and more details in cells, and not much more than 30 years later, a scanning electron microscope, so great improvements in the technology. 1997, Dolly the sheep being cloned. Look that up if you don't know who Dolly the sheep is. And 2000 and onwards, X-ray diffraction, helping us to know really tiny, tiny details of what's going on inside cells. So the X-ray diffraction crystal structure of ribosomes was published in the year 2000, and since has been refined to take it down to a subatomic level. So we know exactly how the machinery of the ribosome is able to work and make proteins. Okay, so what is cell theory? Well, three parts of cell theory. One, all organisms are composed of one or more cells. So that allows for single cellular organisms, and it reminds us that all living things, all organisms have cells. So animals have cells, plants have cells, bacteria are cells, rocks and stones and bricks and water are not alive, they do not have cells. The second part is the cell is the basic unit of structure and organization in organisms. So when we think of the way that an organism is organized, it's the cells of that organism that give that organization. So dividing an organism up into different organs, for instance, so heart or liver or muscle or brain, each of those different tissues or organs within an organism are made up of cells which give that particular organ its characteristic function. Third part of the cell theory, all cells come from pre-existing cells. So if you were listening to the video, you'll remember that there was a disagreement early on about how cells came about, whether they came from pre-existing cells or whether they crystallized out of non-living, out of non-cellular material. Okay, any questions? Pause and think. Okay, so just to finish, I'm going to look at some microscopes and some cells seen through these microscopes. So just to help us to understand how we know about cells. So here is a picture of a light microscope. So this is an instrument that uses light and takes it through a series of lenses which magnify the image and allow an observer to see a much magnified image of something, of a specimen that's on a slide on this part. So a light microscope can allow things to be seen that are not visible to the human eye. This is an electron microscope, and it works on a comparable principle to a light microscope, but rather than using light wave energy, it uses electrons. So an electron source releases electrons that pass through the specimen and get diffracted, get moved aside by structures within the specimen, and then the electrons can be collected, and from where they've been scattered to, an image can be made up by the electron microscope. So here is the sort of image that you can see very easily using a light microscope. These are some epithelial cells, some skin cells from the inside of a cheek, and they're stained with a stain but specific for DNA and viewed under an ordinary lab microscope. So you can see that the stain gives a blue color to DNA within the cells, and I hope that you won't be surprised to see that the DNA is concentrated within the cell. It's inside the nucleus of these epithelial cells. Here are some similar epithelial cells, again looked at through a light microscope, but using a different technique. So this is a staining technique. This is a technique called phase contrast microscopy. So if you like, the light source has been adjusted so it's showing shadows within the cell structure. So you can see that this allows you to see some things that you can't see in this sort of staining view. You can see the cell membrane very clearly. You can see a membrane around the nucleus, and you can also see some structures within the cell. Remember, this is just using light and using a particular technique of the way that the light passes through the specimen. Here's another different technique. In fact, you can see that these are exactly the same cells. This is another technique using a light microscope called differential interference contrast microscopy. And here you can't see some of the details that you see here, but you can see other details. So this is almost like a three-dimensional view of the cell. You can see that some of the structures that you can see here looking quite flat, in this view, it looks as if they're raised up. You can see that the nucleus is somehow higher than the cytoplasm of the cell. And a third technique, this is fluorescence microscopy. So this technique uses a fluorescent stain, that's a stain which emits light, and different stains can emit light of different colors. And these light emitting stains can be attached to antibodies which bind specifically to different structures inside the cell. So here you can see the nucleus showing in yellowy orange and the outer membrane of the cell and structures inside the cell shown in red. Okay, so here is some more fluorescence microscopy. So different dyes showing different colors, different structures within different cells. So if we go back to here, this yellow stain is staining DNA specifically, and the red stain is staining microtubules. So you can see microtubules within the cell, the cytoskeleton, and you can also see that the inside of the cell membrane is lined with microtubules, which give the outside of the cell its structure and shape. Okay, so here is an electron microscope again. So electron microscopes can produce a much more magnified image, and a light microscope can magnify up to about 400 times. An electron microscope can magnify up to about 5,000 times. And again, it has different techniques that allow you to see different features within the specimen. So transmission electron microscopy passes the electrons through a specimen. So it allows you to see details inside the specimen, whereas scanning electron microscopy bounces electrons off the surface and allows you to see details of the surface of the specimens. So here we've already seen this picture. This is the blue-green algae cell again, the Chlamydomonas cell. And this is a transmission electron micrograph. Okay, so you can see that the electrons have passed through the cell and been scattered differently depending on the exact characteristics of the material that it's passing through. So we're able to see this image of what's inside the cell. This is a relatively low magnification. Here is a much higher magnification of just one small part inside the cell. So this is a membrane structure inside the cell, the endoplasmic reticulum, the sort of structure we can see here. And in this higher magnification, we can see details. For instance, the fact that the membrane appears as two dark layers with a lighter center. You'll learn exactly why that is later. Okay, so a membrane bilayer we can see here. And here is a scanning electron micrograph of pollen grains. And this is what I mean about being able to see surface details. You can't actually see anything inside these pollen grains using this technique, but you can see the outside shapes of these tiny, tiny pollen grains, pollen from different plants, each having different characteristic shapes. So some tiny little grains here, some bigger ones. You can see this looks like sort of a honeycomb structure on the outside. Here is a structure with a sort of slit down it, each one from different plants different, and you can see beautifully the detail using scanning electron microscopy. So in the same way, this is a scanning electron micrograph of mammalian cells growing in cell culture. So these are cultured cells outside a living organism. So again, the electrons bouncing off can allow you to see the way these cells are growing and interacting with each other, even though they're being grown totally artificially in a cell culture. Okay, any questions on that? Just pause and think anything you might want to know. Okay, so that's all that I've got for you in this video, except just to mention textbooks that you might want to go to learn more. So my favorite textbook for learning about cell biology is called Molecular Biology of the Cell. I'm a molecular biologist. I like to think of things from a molecular point of view. And this textbook takes that approach, and it has some really excellent illustrations that helps you to visualize what's going on inside the cells. So I will be using this book a lot. You will see a lot of illustrations that I've taken from this book as we go on. So you can find physical copies of this book in the university library, and it's also possible to download soft copies of the book. Leninger is the standard biochemistry book for the biochemistry course you're doing. So Leninger has been around for a long time. It's quite an old textbook. It's been through a number of editions. So the university library has a lot of physical copies of this edition, which is quite old, from 1988. But many of the things you're learning about, we knew about perfectly well in 1988. So for instance, chapter 2 in Leninger covers cell biology quite well. We might know more details of the structures, but for basic information, if you want to read a physical copy of a physical book, there are lots of copies of Leninger in the university library. If you want a more up-to-date copy of Leninger, this one is available. You can find it as soft copy, and chapter 1, section 1 is called Cellular Foundations of Biochemistry. So that will look at cells from a point of view of a biochemist. Finding out information is so easy if you've got access to the internet. The easiest thing to do is just to use Google and put in whatever you're interested in. For instance, I used the search term protein here, and here is the Wikipedia article on protein. I like Wikipedia. It has a lot of good information. Most articles are reliable, but the only downside of Wikipedia is that anybody can edit all the articles at any time. So if you happen to be unlucky and somebody has entered some bad information into Wikipedia when you happen to read the page, you might read that bad information, but very, very quickly, knowledgeable people will correct that information and return the article to being helpful and accurate. Another site I'd like to mention is called Citable, which is made by the scientific journal Nature. So www.nature.com Citable. It's got a lot of good articles in it with good illustrations covering lots of topics in molecular biology, cell biology, genetics. If you just do a Google search, you will probably find a lot of information that's good and will be very useful for you. However, you might also find some web pages which are uninformed or misleading or not really talking about what you want to know about. So be careful and be sensible about the pages that you look. A good hint is to look for pages that come from universities. So these are going to be articles put onto the internet by lecturers who are teaching students this sort of material all over the world. All medical students everywhere in the world learn biochemistry. So there is a lot of material, a lot of teachers teaching biochemistry. So look for web pages with the end bit will be .edu for education or ac for academic or gov for government. I've said here avoid personal blogs because even though there are some excellent personal blogs done by scientists describing and discussing things that they find interesting, sometimes they are scientists who have non-mainstream views and want to find a way of convincing other people of something that's not the mainstream view. So yes, read them if you're interested, but be careful. Don't take it that everything you see on the internet is correct. Okay, so that's all I've got for you. I'm going to finish this video now, and hopefully you're going to be able to download it and watch it and get started on cell biology. So goodbye until next time.